but instead determining for each patient which therapy is going to work best in that specific disease, and then use that specific treatment for that person on a case-by-case basis. So why do we need precision medicine? So I'm going to use cancer as an example, but the story also applies to other diseases. What makes different cancers unique? So first of all, cancers arise in many different anatomical locations, prostate and breast being the most common, but there are many different organs in which cancers arise, and even in the same organ, there's different types of cancers. It's not just where they arise, but also where they come from. So cancers generally are known to come from genetic causes, but there are vast differences in the number of mutations that are underlying a given cancer. On the right, an example is melanoma. There are upwards of 100 mutations per million base pairs are common. On the left are examples like thyroid cancers, where there's typically only one mutation per million base pairs. So there's vast differences, not just in the type of cancer, also among individual patients that have the same type of cancer. So each of those dots is representative of a single patient. And you see, for example, in melanoma, some patients have 100 plus mutations and some have less than one mutation. So the genetic causality here can be very different. And it's not just the number of mutations, it's also the types of mutations. So some, like P53, is a very common mutation that occurs in many cancers, whereas BRAF and others are much more restricted to one or two types of cancer. So the diversity in the genetic causes is also reflected in a significant diversity in responses to cancer treatments. There are about 150 approved anti-cancer agents. Most of these have no good predictive marker of response, and so most patients receive what's called standard of care therapy. There's a certain percentage of patients that respond. In some types, like ovarian cancer, it can be 80% or higher for initial response. And for other types of cancer, like triple-negative breast cancer, the response rates are around 30%. And we don't usually know why certain patients respond and others don't. And even for targeted therapies, like those used in melanoma often, even patients that have a specific mutation that have been confirmed to have this mutation, when they receive the kinase inhibitor that targets it, the response rates can sometimes be high in certain cancers. They can be much lower in others. So even if the genetic picture is pretty clear, the actual response can still vary greatly. And so there's clearly more going on than just at the genetic level. And it's becoming more and more clear that other factors downstream of the genetics can play an important role in drug response. So I've mentioned the complexity at the genetic level, but it turns out that's only the beginning. So cancer starts at the genome, but there's other layers of complexity at the epigenome, at the transcriptome, at the proteome, at the metabolome, and all those different factors affect the phenotype. And that's what we're really interested in, is to see the actual drug phenotype, to see how the tissue responds to an actual drug. The tumor microenvironment plays a huge role in this. On the right side is an image of what we think a tumor looks like on the inside. The pink cells are tumor cells, but you can already see there's many other types of cells. There's immune cells, stromal cells that may secrete fibroblasts, and that can affect. The fibroblasts secrete growth factors, and those growth factors can render certain tumor cells unresponsive to drug treatments that they otherwise would be responsive to. So the complexity is mind-boggling. Going directly to the phenotype is what we felt is an approach that's that's worth taking. It integrates a lot of the complexity downstream and lets us look directly at the drug effect. So we decided that we wanted to test many different therapies in the body, directly in the body, but we can't give many of them at full dose because they're toxic. And so one of the things that we came across is this FDA initiative on microdosing. It's part of the Critical Path Initiative that was meant to address long timelines and growing expenses in drug discovery. And one of the parts of this initiative is what's called phase 0 microdosing. So that allows you to administer very small doses of new drug compounds into the body, and the FDA thought that this would be a good way to learn about things like biodistribution, pharmacokinetics of these drugs early on before they have to be optimized for safety and other factors. So our idea was, let's use this not to look at PK, but to actually look at efficacy of the drugs, and to do this right inside the tumor for all the reasons we talked about, so to look right at the phenotype. So now the question becomes, what's the best way to get these drugs into the tumor in a controlled manner? The way that's most used in the clinic to access the tumor is the biopsy. And so the vast majority of tumors or the vast majority of patients at some point receive a biopsy. It's minimally invasive, it's rapid, it's relatively safe in the vast majority of tumors. So this seems like a great way for us to actually get the drugs into the tumor in a local way and in a way that integrates well with what's already being used in the clinic. And so to quickly summarize why we think we need an engineering approach to precision medicine. So the first reason is we want to test the phenotype, and we want to deliver drugs in small amounts directly into the native tumor tissue and examine what happens there. And so the microdosing concept allowed us to do that. And the biopsies provide the mode of access by which we can get the drugs into the tumor. And so our approach is to use little microdevice constructs that are delivered through a biopsy needle that can hold a large number of drugs and can release them locally in the tumor in a very controlled manner. So we have, there's an actual image of a early prototype shown. So the basic idea is to take a biopsy needle, to put this construct right in the tumor. It remains in the tumor for about a day. During that time, the drugs diffuse out into separate little regions of the tumor. After the incubation period, we come in with a coring needle and core out the device with surrounding tissue, and then we process that tissue in the laboratory, do standard pathology, histology analysis on the sample, basically cut through that entire specimen layer by layer. Thank you. Thanks. And then we get individual sections, many sections per reservoir that we can then use to understand the drug effect, to stain it using IHC and other markers to understand how the drug is acting. So I want to show a little bit, before we get into the data, I want to show a little bit about what we did to take the original idea into what's now a clinical stage device. So the original idea was basically a two-dimensional stamp that has individual components to it, and it's pressed into the tumor, and then drugs diffuse into separate regions. But when we started to think about how to actually get this into tumors, and we realized that we wanted to use biopsy needles, we basically turned this into a cylinder design where the mantle of the cylinder is being used as a place for the reservoirs, and it allows us to place a great number of these into a fairly small construct that then fits nicely into the biopsy needle. some of the early prototypes were then small cylinders that had a relatively small number of reservoirs. We tried some different filling methods for these, and through an iteration process, we arrived at the current prototype, which is optimized to penetrate into tissue. It has a barb at the end that keeps this locked into place in the tissue. And then the spacing and formulation of the reservoirs are optimized for the largest number of wells in the smallest possible design. We've also scaled up the manufacturing efforts, so these are made using CNC micromachining, and we have robots that can fill the little reservoirs with individual drugs. So next, I want to talk a little bit about the process of how these microdevices actually work, how they deliver drug, and then looking at the effect of the drugs and putting it all together. So I want to start out by talking about the drug delivery aspect. So these are examples of different drugs that are being delivered here. What we're basically looking at is a cross-section of tissue. This is a cross-section of the device. It's a cylinder, and there's a port on one side, and the drug, in this case it's doxorubicin, is being delivered out into a small confined region of the tumor. It's being detected by autofluorescence. These are examples of other small molecule drugs, kinase inhibitors, sunitinib and lapatinib. We can also do this with antibodies. This is cetuximab. And we use mass spec MALDI imaging to detect drugs that are not fluorescent. We can also do combinations of drugs. You can do two drugs from one reservoir, or do adjacent reservoirs where the drugs then overlap at a given time point, and you can look at effects such as priming. And you can also look at time effects. You can release reservoirs also at different times based on formulation. So it's also a way to understand drug diffusion in tumors. But for us, it's really the first part of how these devices work is to deliver these small depots of drug into the tumor. So as many of you know, the effect of cancer drugs is dose dependent. So one of the important parts is to control how much drug is being released into the tumor. We want a concentration of drug that's representative of what you can achieve systemically in a patient. So we use different techniques for controlling the local release of drug. It's mostly based on matrix formulation. So the drugs are formulated in a polymer matrix such as PEG or PLGA or others. And many of these drugs have different chemical properties, so they're taken up into tissue at different rates based on size and solubility. So we use these polymer matrices to really control and standardize the rate of release of drugs into tissue. This is an example of a systemically dosed tumor here. So the animal in this case is dosed through a tail vein injection. We take out the tumor, and we see this range of values of drug distribution, and we can recreate that on the actual device release. So really try to match what we get in systemic dosing in the local release from the device. So the next aspect of how these devices work is to understand what the effect of the drugs is. So we basically take those exact same sections, and then we stain them using immunohistochemistry for drug effects. So Clef caspase-3 is a marker of apoptosis. So these brown cells here are cells that were exposed to the drug and are dying as a result of exposure to the drug. We get many different sections from a single reservoir, so we can stain every drug for multiple markers and really understand things like target inhibition, proliferation, and those effects for every single drug that's on the device. And we basically score these images using a standard algorithm and then get a number. In this case, we're looking at apoptosis, so we count apoptotic cells and count the rest of the cells, and then we arrive at a number that we call the apoptotic index. and it gives us a measure of the efficacy of a drug. So one of the really cool things about this thing that I'm still fascinated by is that it allows you to do intratumor pharmacology in a completely new way. So you can understand the effect of a specific drug at different concentrations. You can understand what doses does it take of a certain drug to reach a certain level of target inhibition, of killing effect, etc. So it really allows you to understand how effective a drug is in a tumor, not just in a yes or no manner, but really specifically ask, what do I need to dose this tumor with to get a certain level of effect? You can also look at the kinetics of drug action. It's also a nice feature of this to understand how fast the drug is acting. So one of the key questions in using this is whether the local readout that the device produces is actually predictive of the systemic sensitivities of the tumor. So we've tested this in a large number of different models, and then we've tested those same models systemically, and we really see this one-to-one correspondence between how tumors react locally to drug. In this case, it's a high responder, a medium responder, and a low responder, and that exact sensitivity is recapitulated on the device from what you get when you dose these animals systemically. We've also tested this for targeted agents, in this case, in different tumor models. This is a BRAF mutant one that responds highly to a BRAF inhibitor, and also looked at combinations where the added effect of the, in this case, the HER2 inhibitor is expected based on the choice of model. And then we've tested this in a number of patient-derived xenografts. So the idea is this is a triple-negative breast cancer tumor that we grew in mice, and then we test all these standard of care therapies that these patients would receive in systemic studies, and then we test those locally on the device, and we get this one-to-one correspondence. So in essence, the effect that we read out locally is significant. It shows the inherent sensitivity of the tumor, and we can use it to base systemic therapy decisions on. So the goal, or one of the goals of my work, is to move this away from a test that does yes or no questions on the efficacy of drugs to really be able to do in situ systems biology. So I want to show an example of what I mean by that. So this is a signaling pathway for estrogen receptor positive breast cancer, and there are many different signaling nodes involved that all eventually lead downstream to proliferation or apoptosis or priming. And there are many different drugs available in breast cancer, and it's a difficult challenge clinically to know what do I need to combine in terms of different drugs to get a durable response. Often patients have responses, but then they relapse. So to find the optimal therapy right away among the many different drug choices is a real unmet need here. So the approach that we took is, instead of focusing on one or two or three different signaling nodes, like is often done in systemic studies, we wanted to look at all the available drugs that we could possibly use and test all of them, and to look at combinations of 2 agents, combinations of 3 agents, and the way we do this is a pretty streamlined process. So it's basically using a small number of devices. They're placed right into the tumor. We then core them out, and we get the devices with surrounding tissue. We can then implant them into a paraffin block. We slice them all in parallel, and then get our tissue sections for every level for multiple devices. For all the devices that were basically in this tumor, we look at them at the same time, so we can get many therapies tested in a single tumor. In this case, an animal, but we think this can be done in patients too. I'll talk more about that later. So the goal of this is to, one, identify the most effective single agents and combinations. So as you can see, the single agents, there's different sensitivities here. Once you start combining 2 or 3 different agents and then going into 3 agents at once, blocking multiple of these nodes is a way to success. It's also a way that's being pursued clinically. But one of the cool things is not just to look at which drugs work, but what is synergistic and what levels of a given drug. What's the smallest possible level of a drug I can dose with to get a certain threshold effect? Because clinically often patients are dosed at the maximum tolerated dose, and we don't ask, does it have to be that high, or can we actually go lower? So in this case, we found out that the PI3K inhibitor and the AKT inhibitor, even though they're not terribly effective on their own, when we combine them, we can achieve a very high synergy at drug levels that are actually much lower than what patients typically get when we dose them systemically. So that was an example of a static measurement. We can also do dynamic measurements where we test the same tumor multiple times over different time points during a treatment. So in this case, we took a BRAF mutant melanoma tumor, and we dosed it with the standard of care, a BRAF inhibitor called vemurafenib. And this is a drug that's been known to have very high initial response rate, but resistance rates are extremely high. Most patients relapse in less than a year with more aggressive tumors. So one of the questions has been, what does the BRAF mutant tumor do? What are the escape mechanisms that the tumor uses when the primary pathway is blocked? So we were able to look at this in a functional way and basically test every single one of these targeted agents that represent different potential escape mechanisms of the tumor, and test each of those before treatment, during treatment, and at the end of treatment. So you can really understand the escape mechanisms, what the tumor does when its primary pathway is blocked, what are other pathways that are overexpressed in response to the primary pathway being inhibited. And this is, we identified, again, the PI3K pathway and the EGFR pathway as 2 upregulated measurements. And it's important to remember that the genetics of the tumor stay the same. This is a relatively short-term measurement, but even in these short time frames, the adaptive mechanisms can be significant, and it's what we observe in the clinic. And we think that finding these adaptive mechanisms and adjusting therapies so patients can, in addition to the BRAF inhibitor, then receive a PI3K inhibitor or an EGFR inhibitor or both, would greatly improve response. So just to recap the technology part a little bit, it's a very short-term implantation that goes through a biopsy needle. It's a large number of drugs we can do in a single tumor. We have very precise control over the location and the concentration of the release. This works for small and large molecules, including proteins and siRNA, and it lets us evaluate the effect of concentration and the timing of synergies, basically. So the next part of the talk is about our clinical work. So we've started to translate this into a clinical trial. One of the key considerations in the clinical trial is safety, and we think that this is really, we've tried to design this in a minimally invasive and safe manner from the start, but it was also deemed non-significant risk by the IRB. And the reason for that, I think, is that, one, the drug amounts that are released into the tumor are very small. They're about 1/1,000,000th of what you would give a patient systemically. And that amount is actually largely removed when we core out the tissue with a biopsy needle. So there's really no remnant of drug. It was very little to begin with, but even that is taken out. So from a toxicity perspective, there's virtually no risk to the patient. The device constructs are made out of a long-term biocompatible plastic that's used in joints and other implants, so it has a long-term safety track record. So the real risk factor here is just the biopsies, and in some tumors, maybe biopsies aren't used as often, such as lung cancer, but in most tumors, it's deemed a pretty safe procedure that the radiologists are pretty happy to perform most of the time. So the IRB actually approved this as non-significant risk, so it did not require us to get FDA approval before the trial. And so the study that we have ongoing now is a first-in-man clinical study in breast cancer. We chose triple-negative breast cancer as a good place to start. These patients receive neoadjuvant therapy now, which means they receive therapy before surgery in order to shrink the tumor. And it's typically 2 cycles. It's AC and then TP. So we can administer the device before therapy, so it's on the non-treated tumor, and we test all of the standard of care therapies. We're also testing a few second-line therapies as potential options for later on. So the device is implanted, it's taken out the next day. The patients then go on several weeks of the first-line therapy. There's a research biopsy that's going to allow us to correlate the device effect for those drugs alone. And then they go on the next part of therapy. Again, this is all standard of care. And then the tumor is resected at about 6 months. And then we're able to read out whether there is a correlation between the readout of the device and the systemic response to those drugs in the patient. And this trial is enrolling 12 patients. We've had 3 thus far. So the endpoints are both the technical feasibility and safety, but also the correlation between what the device reads out and what happens to the patient systemically. And on the right is actually an image of a patient tumor with one of the devices in it under ultrasound, which is the mode of implantation and retrieval. So breast cancer allows us to use larger coring needles that other cancer types don't have. So one of the key challenges now is to use smaller coring needles that have a more precise cutting mechanisms. So one of the things I'm working on now is to design the devices with little guide wires on it. We then have stylets that go over the guide wire and provide a very localized and very precise cutting of these tissue cores right around the device. So this also requires a end cutting mechanism because you have to separate the tissue at this end from the rest of the tumor. So we designed these little end cutters that are nitinol tubes that have a shape memory effect that actually close on their own as soon as they protrude past the tip of the coring needle. So it's a nice way to get very precise specimens to really separate them from the rest of the tumor. And again, you can see this nicely on ultrasound, the actual device with the guide wire, and then you see some examples of those cores that were taken from live tumors in this manner. So this allows us, in theory at least, and we're going to test this in the next trial in ovarian cancer, to have much smaller coring needles and a very precise method of retrieval that will hopefully be useful in many tumor types beyond just breast cancer. So next I want to discuss a few of the current research applications for the microdevices. And I've picked a couple examples of basic cancer biology. So we have one of our collaborators, Matt Vanderheiden at the Koch Institute at MIT. is interested in understanding tumor metabolism. And one of the hypotheses that they've been working with is that certain tumors, in this case KRAS mutant tumors, break down protein to proliferate as a fuel source, basically. But this has only been observed in vitro. No one's ever seen this happen in live tumors, in live animals. So what we were able to do is to examine that using the microdevices, releasing protein in the tumor, and then live imaging the effect. And what happened is, so basically, we put the little devices into a mouse pancreas, and they released what's called DQ-BSA. It's a self-quenched albumin. So when it's not cleaved, you do not see it, and when it gets cleaved, it becomes fluorescent. So we were able to see specifically in the tumor tissue that this cleavage takes place. So that was the first clue that something's happening here in the tumor cells that's not happening in the normal cells. So next, we used live multiphoton imaging in a tomato-labeled tumor, and again, this albumin breakdown. So the albumin's in green. This breakdown only happened in the actual tumor cells. So this is live imaging in the mouse tumor. We're able to see this catalysis of the protein happen locally inside the cells. So the next thing we wanted to do is what happens if we inhibit a process called macropinocytosis, which is used to transport these proteins into cells. What if we inhibit that? So we delivered this compound called EIPA locally into the tumor. It's detected here by MALDI mass spec tissue imaging. And then we looked at the distribution of amino acids in the tumor, because if the tumor breaks down albumin, that results in amino acids. And what we see is if we block this process called macropinocytosis, then we also block the production of amino acids, and it was a clear signal that the tumor cells are using these proteins that they're breaking them down and that they're using them as a fuel source. As a control, the choline is still intact in all these cells, so the cell's structure and shape is intact. So the significance here, the big significance, is that we can actually watch this process of protein scavenging live and see it in the tumor. It's never been seen before in this manner. And the second research application I wanted to show is studying migration of invasive cancer cells in situ. So this is a collaboration with Frank Gertler, also at the Koch Institute at MIT, where the hypothesis that they've been working on is that this specific subtype of tumor cells called MENA-invasive is attracted to fibronectin gradients and uses those as a path towards migration and towards metastasis formation. And so we released fibronectin inside the tumor. Again, this is not something that you would be able to do systemically, but using the microdevices, we could just deliver them right into the tumor. And what happens is that these tumor cells build up these collagen fibers, and they use those collagen fibers as a way to migrate towards the fibronectin. So for them, again, it was a nice confirmation that some things that they've been seeing in vitro are actually happening in vivo. And there are other examples of those fundamental type applications where we're able to do things that aren't that easy to do for biologists or cancer biologists by other means. And they've been able to do this through the microdevice by releasing things in the tumor that you wouldn't ordinarily be able to administer systemically. So the last thing I want to talk about is the vision for what we call lab in a patient. So I've talked about the delivery aspect. We deliver things into the tumor. We can read out what happens only after we take out the tissue, and then we analyze it. So the next step is to actually do the analysis part in the tumor, to use methods of detection that allow us to watch what happens to tumors when you treat them with the drug. Why are certain tumors responding and others don't? So to really do this in situ in the tumor and to watch it happen in real time. So this next generation device has optical fibers that are accessing the drug delivery ports of the device. There are multiple fibers looking at a single reservoir. They're looking at the tissue around it. The entire device is still fairly small. It's about 1 millimeter in diameter. And this particular prototype has 12 reservoirs and a fiber bundle that comes out the end. It can be implanted with a 16-gauge biopsy needle, so still fairly small. And it allows us to perform multiple optical imaging modalities on the same device. So we can use, in this case, we can use optical coherence, Raman, and fluorescence testing for every single reservoir, basically in real time. So this eliminates the need for a coring biopsy, and it also has, the benefit of you only have to leave this in the patient until you see an effect. You don't have to leave this necessarily in overnight. You can envision a scenario where a patient receives the device in the morning and then in the afternoon already has their results and knows, and the doctor knows what the tumor sensitivities of that patient are. So there's an example of this is shown on the right here. This is a device reservoir that's filled with doxorubicin. The doxorubicin was released into a confined region of tissue. This is all live imaging in an animal tumor. And then we gave a systemic contrast agent. In this case, it's an apoptosis dye that binds to apoptotic cells. And you see this nice aggregation of the apoptosis dye in the region of drug distribution, showing that this drug is quite effective. It's causing a lot of tumor apoptosis. And we envision doing this for many drugs in parallel on the same device. I talked a little bit about the other modalities, so it's not just the fluorescence, it's also optical coherence tomography in the middle here, which detects a change in scattering in the tissue. So when cells turn apoptotic, they tend to undergo a change in shape, and this shape change results in a change in scattering, and we can use OCT to detect that. Raman spectroscopy is also one of the modalities that we're using. This is actually a signature of the Raman signal that corresponds to DNA damage. It's a great measure. It turns out to be a great measure of apoptosis, and we see this very high correlation. On the right, the corresponding Raman signal. On the left, cleaved caspase 3 staining for apoptosis. So this is a really a nice benefit that you can use multiple modalities to get a more complete picture. And again, to do all this in real time while the device is inserted in the tumor. So last, I wanted to talk a little bit about the longer-term vision where we think this can be used in the future. So the first part of the device is basically the same as the current device. You can imagine testing many drugs at once, but the longer-term vision is really to provide a way for the physician to manage chronic disease and to manage longer-term diseases like cancer, where the tissue changes over time, where it changes during treatment, and you can trigger. In this case, you can trigger the release of drugs at different time points. Let's say you treat the tumor systemically, and you realize that it's growing again, it's relapsing, it's recurrent. You can then trigger another round of tests, and then you choose the optimal therapy at that time point. And we saw that tumors change during treatment. So you pick the new best therapy in this changed tumor, and then you repeat the cycle of test, treat, monitor, because you can use those optical modalities to also monitor the response of the tumor over longer time periods. So the real vision is a longer-term implementation where you're alternating probing the tumor, identifying the best therapy at a given time point, monitoring overall response over longer time periods, and then repeating as necessary. So one of the there was a comment in Nature that discussed our technology, and the image that they chose is this person that's looking inside the tumor with a flashlight and trying to see what's going on. So I think it's a great image because it really gets to the core of this vision. We want to see and understand how cancer and other diseases, how they work, what their response is to treatment, and we really want to see it right at the site of disease, where the disease is interacting with the drugs and where the response matters. And we ideally want to use that information to find the best therapy, to find it for every patient in a very personalized and precise way, and really use this engineering approach as a new paradigm then for precision medicine. So with that, I want to thank my mentors, Bob and Michael, my team of technicians, Alex, Matt, and Chris, my funding sources, and the great list of collaborators I have. And I'd be happy to take any questions you have. Yeah.
I want to follow up on a question that I guess I asked you earlier today. So this is the question of whether or not the information that you learned from local intratumoral delivery could be translated to systemic delivery. So you showed some data there that was pretty convincing, just comparing the results of your device and analysis of your device outcomes to systemic delivery. And I think those were all small molecule drugs.
Yeah, that's right. Yeah, I
So
can jump back there.
One
Yeah.
of the things I'm curious about is there's an increasing prevalence of biological therapeutics, in particular, for example, antibody therapeutics that have higher affinities for their target than chemotherapeutics. And one of the, I guess, just design considerations in applying those drugs is tumor penetration and how the affinity of the antibody to its receptor correlates with tumor penetration.
Yeah.
So you mentioned that you've actually done some work with cetuximab and maybe other biologics. Could you comment on how local intratumoral delivery of those high affinity biologics might correspond with systemic delivery?
So I completely share your concern about transport into tumors. I think for some of these larger molecules, it is a big concern. And there have been some studies that show that it could actually be a mode of resistance in and of itself, that the drugs aren't getting into tumors at sufficient concentrations. One of the neat things about the way we read out results is you can look at many different things in parallel. So you don't have to just look at apoptosis to understand what the drug is doing. You can also look at target engagement as long as you can read it out locally. So that's could be a better measure for some of these drugs than a more downstream effect like apoptosis or proliferation, especially when you're thinking of combining multiple of these that might attack different pathways. All of them might not kill cancer cells on their own, but when combined, they would. So to get these more specific readouts, like target engagement or inhibition of a specific kinase, can be a more informative readout for some of these drugs than something more global like cell killing. Yep.
So there are obviously advantages to going smaller, being less invasive. What are the primary limitations? Are they the morphologies of the cells, or the amount of dose you can deliver, or the fabrication limitations in terms of why smaller?
So why it's better to be smaller, you mean?
How small can you go, and what limits that?
Oh, so one of the limiting factors is the spacing between adjacent reservoirs. So the drugs, after they're released, they do. If you wait long enough, they're going to overlap with other drugs, and then reading out an individual effect for a given drug becomes impossible. So that's one of the limitations, I guess, and that's on the order of say 200 to 300 to 400 microns per drug per day. So if you have compounds that act over several days, like if you're looking at, for example, RNAs or epigenetic modulators that might require longer time periods to actually show an effect, then you do have to space reservoirs out further so they don't overlap. But for most of the cytotoxics, of the kinase inhibitors, the 1 day time period is actually sufficient, and that allows you really to put a lot of these reservoirs on a small construct. The other consideration is getting a robust enough signal. So you can imagine if you make the reservoirs ever smaller, and at some point there's only going to be a handful of cells that you're interrogating. So actually, in some ways, you find a sweet spot between making it as small as possible, but also have every reservoir be large enough so the number of cells isn't too small. So maybe you're looking at several hundred cells, or even a couple thousand cells that are exposed to a given drug, and that's going to give you a much more robust signal of whether that drug actually works.
And in terms of the time dependence, do you expect that to be linear or diffusion square root of time?
So it's a little bit, because it is a limited amount of drug, it's actually kind of travels as a wave through tissue, as a peak that goes out in more or less a linear way, but it does broaden as it goes further out. So it's more that pulsatile delivery rather than a continuous delivery.
So it's a diffusion here.
Yeah. Mm-hmm. And the drugs are formulated in their matrix, so we can control the rate of release into tissue. Yeah.
Actually, not bad.
So do you notice any changes in the tumor physiology? It seems like the placement of this device may do some micro damage to the tissue. Because you might see some enhanced macrophage activity in that region, right, where you're doing all your measurements. So it might skew your results. Have you looked into that kind of thing?
Yeah, so it's typically the damage to tissue is restricted to the first few cell layers, so to the first 20, 30, maybe 40 microns. But the drugs are diffusing out 200, 300, or 400 microns. So maybe you do exclude those first couple layers of cells, and you typically see some damage there. But the bigger region away from that immediate device-tissue interface is the more interesting one, and you have plenty of cells to look at there.
Similar to that question, how are you confirming that you are putting the device in the correct spot? Obviously, in the xenograft model, you have a huge tumor ball, but say in cancer with the ill-defined margins, how are you confirming that you've actually placed it in the cancer tissue?
Yeah, so in patients, you mean? Yeah, because in animals, it's pretty straightforward. So I'll go back to the example of the breast cancer patient. So the devices have pretty good contrast on different imaging modalities. In this case, we're using ultrasound. And in the patient data that we've seen so far, the margins of the tumor have been easily detectable. The tumors are much larger than the device, so it's easy to know that you're actually in the tumor. If you wanted to place them exactly at the margin, I could see how that would be tricky. But we're putting multiple of them, in this case 3 devices per tumor, and it's been relatively straightforward in this cohort, fortunately. But yeah.
So my question was just about heterogeneity within the tumor environment itself,
Yeah.
and how that impacts your measurements. So at the distal and proximal end of your device, I mean, what kind of impact does that have?
Yeah, I mean, it's a great question. It's really in cancer, especially, a cause of concern, and many doctors are worried about it. That you give a certain drug, it kills some of the cells, but other cells are not being killed, and then the tumor comes back in in a worse way. So in some ways, it's a restriction of every diagnostic. Every time you do a biopsy, you're only taking out a small number of cells, and you're basing your staging or your, even in some cases, treatment decisions on that small sample. For us, the way around it, at least partially, is to put multiple devices into different parts of the tumor. In the breast trial, we have a 6-fold coverage for every drug. So we're able to get 6 data points for every drug from different parts of the tumor. And it's not just a good way to, I guess, to understand the data, but also to understand heterogeneity in a new way. If you can actually now test drugs selectively in different parts of the tumor, and then you can understand maybe why certain regions or even specific cells are not responding to your drug when maybe they should, just given the specific dose of the drug that's present.
So cancer chemotherapy is generally limited by dose-related toxicity.
Mm-hmm.
So I'm just wondering, since you have a method now to get a good reading on effectiveness of different chemotherapeutic agents on the tumor tissue, have you considered putting identical devices into normal tissue to get a control baseline on the toxicity to that particular patient's tissue?
Mm-hmm. So I think one of the goals is to actually move away from this paradigm of always giving the maximal dose, but instead figuring out what dose you have to give to get the desired level of effect, so to actually be able to lower the dose, lower the side effects, and the toxicities. We have not put it into normal tissue. I think in the animals it's a little bit tricky for technical reasons, but and in some cases the toxicities are different too. So, maybe you could put this in the liver and understand what happens to the cells there, but it's not 100% clear on what your readout would be and how you then correlate it with typical systemic toxicities.
So what would be involved in taking this trial design where you didn't have no safety risk,
Mm-hmm.
and changing that to a trial design where outcomes were actually informing the treatment?
Mm-hmm. I actually think it's the next step. So the first step is showing safety, and then kind of technical feasibility. So to actually see that you can get a readout from your sample, that you can analyze the effect of every single drug, and then the next step, I think, is to actually use it to guide therapy, it could be part of an adaptive trial where patients are given a backbone of standard of care, and then the device decides what gets added to that backbone. That could be one way, or it could be in a recurrent disease setting where we really have no idea what to give, and the standard of care options have been exhausted, and you could potentially test a wide range of drugs, with the hope that you'll find at least one or two that show some efficacy.
I guess I think of perhaps a big opportunity here when looking at immune checkpoint antibodies, and have you looked at the potential to evaluate sort of immune cell activation and proliferation in the tumor microenvironment? Is that on your radar?
Yeah, definitely. So we've started actually 2 collaborations. One looking at synergies between chemotherapy and immunotherapy. So figuring out what's the best way to combine them both from a timing and a dosing perspective. And the second effort is to basically look for predictive markers of immunotherapy, because there's really no good way now to predict who's going to respond. The responses in some patients are fantastic, but we don't know why they respond specifically, or certainly don't know how to predict it. So if we could have an impact there, it would be great. Yeah.
So does it appear, based on any studies, that the time scale of just a day is effective enough to actually see if you're getting infected?
So for those, we did 2-day time points, and that actually showed a fair amount of activity. I think we could actually go even a little shorter. Yeah. Mm-hmm.
think that opened up a lot of capabilities. And
Yeah, that's right.
fascinating imaging group here. So any thoughts on that in terms of what are the imaging functionalities are considered to couple this drug delivery monitoring process?
Yeah, so it's one of the things I'm really excited about too. So the ability to image on fibers is important because it's really our only means of access into the tumor. And then the molecular specificity. So are there reliable signals that are used in these modalities that can tell us how the tissue is responding? I'd say those are the two main considerations for picking the optical modalities.
No further questions for now. Let's thank our speaker again.